What I want to talk to you about is some of the biomarker work we've been doing, but thinking about it as potentially providing us some insights into pathophysiology. So while I'm not going to be going deep into pathophysiology, I do want to use this to try and drive some of our thoughts about some of the mechanisms in vascular cognitive impairment. So I know I don't really need to give much background to this audience, but I do want us to think about changing our paradigm. So I think I was raised on this pie chart scientifically. This pie chart existed when I started my graduate school, and this is how a lot of our trainees think still about how dementia exists. And I think of it as a reductionist view. That is, that you have Alzheimer's, or you have vascular dementia, or you have Lewy body dementia. But I think most of the people on this session right now would agree that the real picture is far more murky. And, VCID is, I would argue, probably the largest swath as far as population goes for brain pathologies. Most often it is comorbid with some Alzheimer's changes, although, as we know, that is most certainly not necessary. We also know that Lewy body changes can coexist with these pathologies. More recently, we know that there's a significant overlap of TDP-43 proteinopathy or late. And at least in our Alzheimer's Disease Center, which is a community-based cohort, we have many people that have the quadruple pathologies of amyloid, tau, TDP, synuclein, often on a background of small vessel changes. So understanding how these pathologies contribute to one another is important. Also, from a clinical standpoint, our future treatment of dementia is going to rely on our identification of biomarkers. And I think the biomarkers also afford us an opportunity to forge new research paths. And I'm going to talk to you towards the end about a new biomarker that we're interested in, that we want to try and understand more about the disease mechanisms that it's informing. So the first few slides that I'm going to show you are really derived from the MARC-VCID consortium. I'm sure many of you are very familiar with MARC-VCID. We just entered our 5th year as a consortium. And the overall goal of this consortium is that among these 7 sites that you see in blue here in the US, we are all at the point of cross-validating candidate biomarkers for small vessel disease. These are imaging-based and also fluid-based. And we have one retinal imaging biomarker that some sites are validating as well. The overall goal and the consideration of this is that a lot of us are able to identify, biomarker patterns in our own small cohorts, but once we try to replicate that at other sites, a lot of times that falls apart. And so the goal of the consortium is that that we have candidate biomarkers, what we call kits, and that these kits are being tested and reproduced in the cohorts across the consortium before they advance to being used in settings of clinical trials. So what I want to focus on is the fluid kits. I think this is where we can get some insights into mechanisms And so the first kit is what is being called the endothelial signaling kit. It is a composite of 3 plasma proteins, VEGF-D, placental growth factor, and BFGF. These are 3 angiogenic proteins, and they are measured using the MSD, the Mesoscale Discoveries platform. There is a VPlex assay called the Angiogenesis Panel 1, and that actually has 6 proteins on it. These 3 are a part of that. So we're going to get more data than just those 3, but this kit is a composite of those 3. And it was shown by Joel Kramer's group that cross-sectionally, this kit at baseline predicted accelerated white matter injury and also accelerated cognitive decline. And this is using a composite executive function score. But then also the kit is associated with higher cerebral free water. So this is Charlie DeCarli's free water imaging piece, and then lower brain fractional anisotropy. And that's if you control for the presence of AD by amyloid PET. And so that's kit 1. And just if you just keep angiogenesis in your mind for a minute, we will, I think I skipped one here. No. We'll move to this kit, which is the CSF placental growth factor for small vessel disease. So PLGF was a component of that prior kit that's a plasma kit. This is a CSF kit. The goal of this kit is that the PLGF will be combined with the existing biomarkers of AD and neurodegeneration, being A beta, tau, and NFL. And so our goal overall of this kit is that there will be kind of a stratification kit for trials. And so you would run this 4-piece kit, and this would provide AD, neurodegeneration, and small vessel disease diagnoses, if you like. And allow for group balancing or exclusion for various pathologies. And so interestingly, placental growth factor has shown in MOC-VCID a strong relationship with cerebrovascular disease, and that's separately in our University of Kentucky cohort, and also in the San Francisco cohort of Joel Kramer and Fanny Elahi. And so, as I said, the rationale for developing this kit is that any trial, either for small vessel disease or AD, is going to require confirmation that, number one, the targeted pathology is there, and assurance, where possible, that comorbidities are minimal, so that you maximize the potential of success for that clinical trial. And just shown down here, and I'm going to talk about PLGF data a little bit more, is the relationship that we found. So with increasing white matter hyperintensity volume by MRI, you see that placental growth factor is increased in the spinal fluid. And this is being measured by the Quanterix Simoa kit that they have. And so, you know, when we saw this data, and I think some of you may have heard me talk about this at the AAIC Sydney meeting, you know, we really kind of sat there and went, well, what the heck is placental growth factor doing in the brain, and why is it having this effect? So I went on a rapid learning curve about what placental growth factor really is, and it's a member of the VEGF family. Alone, it is weakly angiogenic. So the VEGFs all exist as dimers. to have their action. And PLGF, when it forms a homodimer with itself, it is weakly angiogenic, but when it forms a heterodimer with VEGF-A, it is an especially potent angiogenic signaling protein, and it binds to VEGFR2. There have been PLGF knockout mice. These mice have impaired angiogenic response in response to pathological conditions. So what is interesting is developmentally, they're normal. There doesn't seem to be any adverse effects of on angiogenesis from a developmental standpoint. But once you induce an ischemia, and this has really only been studied in the limb and in the heart. I've talked to the people that made this mouse, and they haven't really looked at the brain. But when you induce ischemia in the periphery, at least, there's an impaired angiogenic response and a worse outcome. So we are in process right now of obtaining one of these knockout mice that still has a gene product, and so you still get formation with formation of the heterodimer. It just doesn't signal, which I think is probably a more physiologically relevant knockout. So you can see here, we see the relationship of PLGF with white matter hyperintensities, both in the CSF and in the plasma, which is encouraging. The levels, the absolute levels in plasma are lower than what you typically see in the CSF. And so we did some staining. This is human autopsy tissue from an individual. Well, we've done several individuals now who had significant small vessel disease, and then also age-matched control individuals who had minimal small vessel disease. And this is in the white matter in the posterior region, periventricularly, and what you see is there is significant PLGF labeling in the white matter associated with the vessels in the individuals with disease. We are working right now on double labels. The shutdown from COVID really slowed the progress of this project down, but we're exploring what cell types are expressing this. There's definitely some glial looking cells, I think maybe astrocytes, but those perivascular cells, they may be parasites and maybe endothelial cells. So we're working on that. But what you can see here is these same cases. If we took the last plasma draw before the individual died, you can see PLGF is significantly elevated in the plasma, VEGF-A slightly elevated, but not significantly. When we looked at gene expression from matched regions, frozen tissue, we saw increased gene expression of PLGF and VEGF-D. Again, VEGF-A slightly, but not significantly. And then we did a protein expression of PLGF, and again, brain PLGF is significantly increased in those individuals with small vessel disease. We've looked in our hyperhomocysteine mouse model, hoping that this would provide a model that we could study these mechanisms in. The staining, as you can see, is not great. I think there is some elevation of PLGF. Our main issue, I believe, is that the mouse doesn't have much white matter. And so the staining in the human brain, it does seem to be heavily concentrated within the white matter. We don't see much gray matter staining. But in the mouse, we do see elevated PLGF in the plasma of the mouse. Those levels obviously are much lower than we see in the human. Gene expression of PLGF, VEGF-A, VEGF-D all increased, and then brain protein is increased here. So you know, altogether, I think this really does point to some angiogenic dysregulation in cases of small vessel disease. We did a small pilot study with Fanny Elahi from UCSF, and looking at endothelial astrocytic and neuronal exosomes isolated from the plasma of our human participants, really just to see what cells might be contributing to the PLGF we see in the plasma. We did not see any PLGF signal from neuronal exosomes. We did see astrocytic exosomes contributing to the PLGF, and also some endothelial exosomes. But as I said, the gold standard is going to be that double label that we get going to see which cell types are expressing this in the brain. So just to summarize, I think fluids, plasma and serum, as well as CSF, are going to provide an opportunity to identify profiles for neurodegenerative and vascular diseases. And I think if we can really get these fluid biomarkers, it's going to allow us to identify multiple pathologies. I don't envision us, you know, being able to do, you know, 8 or 10 PET scans on individuals. Two of the kits that have advanced to validation in MarkVCID focus on angiogenesis. And I think this really does implicate angiogenic processes in small vessel disease. And this placental growth factor seems to be particularly interesting. It's a component of both of those kits. And it seems to have a role in white matter disease and has high expression in the white matter of individuals with small vessel disease. And right now it looks like astrocytes and endothelial cells are contributing to at least the circulating levels that we see in the plasma. So with that, I'm going to thank my lab who did all the work, and this was us going to a painting class at Christmas. That was our Christmas party. MarkVCID sites who have contributed samples and data, as well as all of my collaborators, and of course the funding and our research volunteers. So thank you for your attention.